Hello students, today we will learn about microbial production of organic solvent. Let's begin from starting. We'll discuss. Now, the commercially important organic solvents are ethanol, acetone, butanol, glycerol. These compounds were mostly being produced in petrochemical industries and little attention was paid for their microbial production. It was in mid 1970 due to economic and political reasons petroleum and natural gas became scarce. Since then, scientists have started looking for suitable alternatives for commercial production of organic solvents. Microbial production of organic solvents utilizing low cost raw materials is considered to be an ideal alternative. Raw material means example wood, cellulose and starch. Now from starting we will discuss about alcohol. So, alcohol chemically ethanol C2H5OH has been produced by fermentation for thousands of years. However, this was mostly associated with brewers and distiller industries. At present, most developed countries produce industries, industrial alcohol by chemical means. The petrochemical ethanol is manufactured by hydration of ethylene, that is C2H4. In the developing countries, microbial fermentation processes are preferred for the production of alcohol. This is mainly because of the cheap raw material available. At the current rate of oil price, it is cheaper to produce alcohol by petrochemical means rather than fermentation. However, large-scale production of alcohol in a given country depends on the political and economic considerations besides the availability of raw materials. With increasing oil prices, many countries now release the potential of alcohol production by fermentation. Now let's know about ethanol as a motor fuel. Prior to Second World War, ethanol was extensively used as a motor fuel. In fact, some motor companies, example Henry Ford, designed some vehicles to run on alcohol or petrol or a mixture of both. Brazil was the first country to produce ethanol in large scale by yeast fermentation, utilizing sugarcane and cassava. This alcohol used as motor fuel is referred to as green petrol. The alcohol used as motor fuel is referred to as green petrol. Brazil started growing alcohol producing plants in a vast area, growing alcohol producing plants in a vast area and installed several fermentation and distillation plants. By 1988, about 90% of the new cars in Brazil had engines that run on alcohol as a fuel. Excited by the success of Brazil, many other countries have also started large-scale production of alcohol by fermentation. Now, let's know about the advantage of ethanol as motor fuel. There are distinct advantages of using ethanol in place of petrol for motor vehicles. Ethanol powered engines can cause less environmental pollution. They produce 60% less carbon dioxide, 65% less hydrocarbons, and 15% less nitric oxide when compared to petrol run vehicles. Thus, vehicles run on ethanol or ethanol petrol mix cause comparatively less global warming and are economic friendly. Another advantage of ethanol used as a motor fuel is the safety. The flash point of ethanol is 3 times higher than that of petrol, 45 degrees centigrade for ethanol compared to 13 degrees centigrade for petrol. Now let's know about the disadvantage of ethanol as motor fuel. At present, alcohol is about twice costlier than petrol in most countries. Since the plus point for ethanol is higher, starting of engines in cold will be difficult. Ethanol may react with alloys containing aluminum and magnesium and may damage the containers. The alcohol used in motor vehicles should be of high purity and should not be allowed to pick up water from the air. A mixture of ethanol with water does not burn readily, and further it also causes corrosion of engines and tanks. Now let's know about gasohol as a fuel. A mixture of about 20% ethanol with 80% petrol, known as gasohol, is now being used as a motor fuel. Some countries like USA, the ethanol used for this purpose should have 100% purity. Otherwise, petrol and ethanol do not form a uniform mixture. Now let's know about production of ethanol by fermentation. As already stated, in recent years, many countries have started production of ethanol by fermentation process. The organism and the raw materials used along with the biosynthetic pathway, the production and recovery process for alcohol. Now we will discuss. First, microorganism. Certain yeast and bacteria are employed for alcohol fermentation. The type of the organism chosen mostly depend on the nature of the substrate used. Among the yeast, Saccharomyces cerevisiae is the most commonly used, while among the bacteria, Zymomonas mobilis is the most frequently employed for alcohol production. Now, let's know about raw materials. There are large number of raw materials that can serve as substrates for alcohol fermentation. They may be broadly categorized as sugary material like molasses, whey, glucose, sucrose, starchy material like wheat, rice, maize, potato, and cellulosic material means wood, agricultural waste. The biomass, starchy and cellulose material significantly contributes for the production of alcohol. Now let's know about pre-treatment of raw materials. Most of the raw materials of alcohol fermentation require some degree of pre-treatment. The actual process depends on the chemical composition of raw material. Raw material, in general, the sugary raw materials require mild or no pre-treatment, while the cellulosic materials need extensive pre-treatment. Now let's know about biosynthesis of ethanol. Biosynthesis of ethanol. The pathway for biosynthesis of ethanol. Glucose gets broken down to pyruvate by glycolysis. Under anaerobic conditions, that is in the absence of oxygen, pyruvate is converted to acetaldehyde by the enzyme pyruvate decarboxylase. Acetaldehyde is then reduced by alcohol dehydrogenase to form ethanol. It is observed that under aerobic conditions with excess glucose content in the medium, the microorganisms grow well without producing alcohol. However, under anaerobic conditions, growth slows down and alcohol production occurs. Regulation of synthesis. Let's know about the regulation of synthesis. Ethanol at high by concentration in the medium inhibits its own biosynthesis. It is particularly observed when yeast are the fermentation organisms. It is generally seen that growth of yeast ceases at 5% ethanol concentration, that is volume in water. volume per volume in water. It is striking to note that yeast are sensitive to inhibition by endogenously synthesized ethanol and not to the ethanol added to the medium. Some biotechnologists prefer to use the bacterium Ximononas mobilis since it can tolerate a high concentration of alcohol. Now let's know about glucose to alcohol conversion profile. Theoretically, the 1 gram of glucose can be converted to 0.511 grams of ethanol. In fact, a conversion yield of 95% was observed when pure substrates are used. For industrial grade raw material, the yield is around 90%. It is estimated that for 100 gram pure glucose, 48.5 gram of ethanol is produced, along with 46.5 gram of carbon dioxide, 3.3 gram glycerol, and 1.3 gram of biomass, that is yeast cells. Now let's know about production process of ethanol. Now let's know about production process of ethanol. Ethanol production can be carried out in three stages of preparation of nutrient solution and inoculum and fermentation proper and recovery. Let's know about the systemic presentation. First is raw material. Raw material needed is starch, cellulose, molasses. Then pre-treatment. Pre-treatment means hydrolysis, clarification and filtration. Then sterilization, pre-culture, fermentation. After sterilization, fermentation. Then recycle, that is separation of cell material, recycle to fermentation, pre-culture. Then after separation, distillation occurs, then dehydration, then denaturation and stillage. Then fuel feed fertilizer. This is the last stage. Okay. Now let's know about the preparation of nutrient solution media. The various raw material for alcohol production have already we have discussed. The most commonly used raw material are molasses, whey, grains, potatoes and wood waste. When molasses is used for fermentation, it is diluted with water so that the sugar concentration is in the range of 10 to 18%. A concentration higher than this is detrimental to the yeast. When starchy material like corn, barley are used, they have to be first hydrolyzed by pre-treatment for use as nutrient. This may be done by barley malt, dilute acids or fungal amylases. Example: Aspergillus species, Rhizopus species. Most frequently massing with barley malt is used. Now let's know about molasses. Molasses contains most of the nutrients required for alcohol fermentation. However, ammonium salt such as ammonium sulfate or phosphate are added to the nutrient solution to supply nitrogenous and phosphorus. The pH of the medium. However, ammonium salts such as ammonium sulfate or phosphate are added to the nutrient solution to supply nitrogen and phosphorus. The pH of the medium is adjusted to 4 to 5 by adding sulfuric acid or lactic acid. Now let's know about preparation of inoculum. After selection of the desired organism, yeast or bacterium its isolation in pure form, the inoculum is prepared under aseptic conditions. For this purpose, the organisms are first cultured in flask under aerobic conditions to increase the size of the inoculum, which can be used for inoculation. Now let's know about fermentation proper. Batch formation process was originally adopted in Brazil. Now, at most places, continuous fermentation is used. There are several advantages of continuous fermentation. These include the retention of the fermenting organisms in the bioreactor by separation and recycling, and the continuous evaporation of fermentation broth. It has been possible to increase alcohol production by 10 to 12 fold by continuous fermentation compared to conventional batch fermentation. Now let's know about fermentation proper. Batch fermentation process was originally adopted in Brazil. Now at the most places continuous fermentation is used. There are several advantages of continuous fermentation. These include the retention of the fermenting organism in the bioreactor by separation and recycling, and the continuous evaporation of fermentation broth. It has been possible to increase alcohol production by 10 to 12 fold by continuous fermentation compared to conventional batch fermentation. Industrial production of alcohol is carried out in huge fermenters up to a size of 1,25,000 gallons. The volume of inoculum is usually around 4% of the fermenter. The ideal pH is around 4 to 4.0 to 4.5. As the fermentation proceeds, the temperature rises to around 30 degrees centigrade. It is necessary to use cooling devices and bring down the temperature to less than 27 degrees centigrade. Ethanol gets evaporated at temperature above 27 degrees centigrade. Aeration, that means oxygen supply, is initially required for good growth of the organisms. Later, anaerobic conditions are created by withdrawal of oxygen coupled with production of carbon dioxide. It takes about 2 to 3 days for the fermentation to be completed. The actual time period depends on the raw materials used and the temperature of incubation. As the fermentation is incomplete, the fermentation broth contains ethanol in the range of 6 to 9% by volume. This represents about 90 to 95% conversion of substrate to ethanol. The overall empirical formula for ethanol production is C6H12O6, that is glucose, in after fermentation gives rise to ethanol, that is 2C2H5OH plus carbon dioxide, 2 carbon dioxide, 2 CO2. Now let's know about recovery of ethanol. Now let's know about recovery of ethanol. The cell mass is separated by centrifugation or sedimentation. Ethanol from the fermentation broth can be recovered by successive distillations. By this process, it is easy to obtain ethanol of around 95% for the concentration above 95%. Special technique of distillation have to be adopted. For preparation of absolute 100% alcohol, an azeotropic mixture of benzene, water and alcohol is first prepared. This mixture is then distilled by gradually increasing the temperature. By this technique, it is possible to first remove benzene ethanol water mixture and then ethanol benzene mixture. Thus, absolute alcohol is left out. Now let's know about stillages in alcohol production. Large volumes of waste, which are technically referred to stillage, are found during the course of alcohol fermentation. Attempts are made to fruitfully utilize stillages for various purposes, to use as feed or fertilizers, for converting to single cell protein, to use a fuel for production of methanol. Now let's know about genetic engineering for improved alcohol production. With the advent of recombination DNA technology, attempts are being worldwide to develop new strains of organisms for increasing alcohol production. Some success has been achieved in creating more efficient microorganisms with desired characteristics. Improved production of alcohol. First point is improved production of alcohol. Second is resistance to high alcohol concentration. Third is resistance to high temperature. Fourth is rapid conversion of substrate to alcohol. It has also been possible to improve alcohol production by using immobilized enzymes in reactor systems. Now let's know about acetone and butanol. Both acetone and butanol are good organic solvents. Decades ago, acetone was used as a gelatinizing agent for nitrocellulose in the manufacture of explosives. It is also used in dyestuff industry. Butanol is required for the synthesis of butadiene to produce synthetic rubber. At present, butanol is extensively used in brake fluids for antibiotic recovery, as amines for gasoline additives, and as ester in protective coating industries. The production of acetone and butanol by anaerobic fermentation is almost a century old. This process was carried out by using starch or sugar-rich raw materials, that is potato, molasses, and the organism Clostridium butylicum. After World War II, acetone and butanol became readily available from the byproducts of the petroleum industry. The production of acetone and butanol by fermentation is almost continued in many countries. It is, however, said that being a very simple process, developing countries with cheap raw materials may soon opt for fermentation rather than petroleum-based products due to economic reasons. Now let's know about production of acetone and butanol. Certain bacteria of Clostridium species are capable of producing acetone, butanol, butyric acid, and isopropanol through fermentation of molasses, wood hydrolysates, starch, sucrose, and pentoses. Depending on the strain of the bacterium and the fermentation conditions employed, the relative production of the four compounds varies. For Clostridium acetobutylicum, the fermentation products are acetone and butanol. Butanol isopropanol or butyric acid, acetic acid can be produced by Clostridium butylicum. Biosynthesis. Uh, let's know about biosynthesis of acetone and butanol. The sequence of reactions leading to the formation of acetone and butanol, along with acetic acid and butyric acid. Glucose on degradation from the pyruvate, which then gets converted to acetyl coenzyme A. A part of this acetyl coenzyme A can form acetic acid. Two molecules of acetyl coenzyme A condense to produce acetoacetyl coenzyme A. The latter undergoes a sequence of reactions to form beta hydroxybutyrate butyryl coenzyme A, crotonyl coenzyme A, butyryl coenzyme A, and butyric acid. This butyric acid is reduced to butanol. For the synthesis of acetone, acetoacetyl coenzyme A combines with acetyl coenzyme A to produce beta hydroxy beta methyl butyryl coenzyme A, acetoacetate acetic acid, and finally acetone. Now let's know about production process of acetone and butanol. Acetone and butanol producing bacterium Clostridium acetobutylicum can be stored in the form of spores for 25 to 30 years. The inoculum is built up in several stages. Such inoculum more efficiently ferments sugars to give a high yield. These organisms are also resistant to contamination. The fermentation is usually carried out in corn or molasses-based medium. The molasses medium contains molasses, ammonium sulfate, calcium carbonate, and sometimes corn stick liquor. The fermenters, after sterilization, are gassed with carbon dioxide. After the addition of medium and the inoculum, The fermenter contains are again stirred with carbon dioxide. The fermentation is carried out with a starting pH 5.8 to 6.0 and temperature 34 degrees centigrade. The actual process of fermentation spread over 36 hours occurs in three phases. Phase 1, phase 2. Let's know about the phase 1. Phase 1 is characterized by rapid growth of the organism and production of acetic acid and butyric acid. There is an increase in titrable acidity. The pH of the medium drop around 5.2. Now in phase 2, phase 1, phase 2, phase 3. So now phase 2. In phase 2 what happens? In phase 2 an increased production of acetone and butanol coupled with a decrease in titrable acidity. Third phase. In third phase what happens? In third phase there is a decrease in the solvent production with a slight or no increase in pH. The fermentation process with this reference to solvent production and titrable acidity we now discussed. Now let's know about contamination. Absolute sterile conditions are required for good production of acetone and butanol. Contaminations due to bacteriophages and lactobacilli is a major problem that hinders the yield. Now let's know about product yield. Approximately 30% of the carbohydrate gets converted to neutral solvents in the fermentation. In molasses medium, the ratio of butanol and acetone is 7:3, while in corn medium it is 6:3. The actual production of butanol is influenced by its toxicity to the organism. Butanol is toxic at a concentration higher than 13.5% in the medium. Now let's know about product recovery. Acetone and butanol are recovered through continuous distillation and fractionation. The leftover residue after distillation can be dried and used as animal feed. Now let's know about glycerol. In details, we'll discuss about glycerol. Glycerol is widely used in industry and commerce. It is particularly important as a starting material for the manufacture of explosives. Glycerol is commercially produced by the saponification of fats and oils. It can also be chemically synthesized from propylene or propane. As such, glycerol is not actually produced by fermentation. During the First World War, the Germans could not import vegetable oils to produce glycerol due to British naval blockade. And for this reason, Germans were forced to produce glycerol by microbial fermentation. At present, glycerol fermentation is of no significant exception, theoretical and historical importance. In addition, this fermentation process also demonstrates how to modify the fermentation condition leads to a modification in the product formed. Now let's know about the production process of glycerol. Basically, glycerol is produced by yeast during the course of alcohol fermentation. However, in the normal process, the quantity of glycerol formed is very low. Addition of sodium sulfate blocks alcohol production and diverts the pathway for the large-scale production of glycerol. Sodium sulfite reacts with carbon dioxide in the medium and gets converted to sodium bisulfite. The latter combines with acetaldehyde, an intermediate in ethanol formation, to form acetaldehyde sulfide complex and thus blocks alcohol synthesis. NADH generated in one of the earliest reactions of glycolysis is utilized for glycerol production instead of ethanol formation due to inhibition. The fermentation is run for 2 to 3 days. The yield of glycerol is in the range of 20 to 30% of the sugar used. This yield is about half of the theoretical value due to the fact that ethanol fermentation cannot be completely inhibited. Further, the recovery of glycerol from the fermentation broth is incomplete. Now let's know about glycerol production by alga. A novel fermentation process for glycerol production by the alga Dunaliella salina has been developed in Israel. This organism, commonly found in salty lakes, synthesizes glycerol within the cells to balance the high osmotic pressure in the surroundings. Thus, the higher is the salt concentration in the surroundings, the more is the intracellular glycerol production. However, when the surrounding salt concentration is suddenly reduced, glycerol is excreted into the medium. This principle is successfully exploited for the production glycerol by desalina. Now let's know about glycerol production by Bacillus subtilis. Bacillus subtilis is normally an aerobic microorganism. It is capable of converting glucose to glycerol, ethanol, lactic acid, butanol, diol, etc. But the production of glycerol under aerobic conditions is very low. However, under anaerobic conditions, the production of glycerol by Bacillus subtilis is quite high, and the yield of glycerol comparable to that obtained by yeast fermentation. Finally finished. Any questions regarding this, you can ask me in the comment box. I will definitely answer the questions. And to know more updates about biotech topics, and if you are preparing for CSIR UGC net, you can follow my videos and subscribe my channel for more updates. Thank you.